Okay. Second semester master students, please fill the Google form I shared. Okay. Master students, second semester, please fill the Google form. Okay. And please write your name and UID in the chat box. WhatsApp. Okay. You sent to me on official mail. Which email? FMLT. 1 second. Let me class. Okay. Sir, I don't know. It's talking. Okay. Students, please turn on the cameras. I will take screenshot. Turn on the cameras, everyone. Just for 1 minute. Okay. Thank you, Sangeeta, Akash, Adit. Okay. Thank you. You can close now. Okay. So, okay. Now let us see about electrophoresis. So in the previous lecture, we were discussing about chromatography. So as we know that chromatography is used for separating the molecules, but there is a limitation to the chromatography. Some of the limitations and some of the drawbacks of chromatography are chromatography is a time-taking procedure and it requires more patience and attention while we are performing this chromatography. So if you do any mistakes, the chances of getting errors is high. Okay, you need to have a right composition of the solvent, mobile phase, the pore size of the this capillary, this filter paper, and also you need to consider the type of dye you are using to visualize the molecules. Okay, so all these things considering it will take too much time and it's a very sensitive procedure. So these are the few drawbacks of the chromatography. And one more problem with chromatography is it cannot separate the molecules based on their charge. Okay, so as we know that all biomolecules have certain electric charge to them. Okay, that depends upon the type of amino acids the molecules have. Okay, we know that amino acids or basic amino acids, and they have some electric charge to them. So depending upon this electric charge, we can able to separate molecules, but that cannot be achieved with the help of this chromatography. So we need an alternative method that can facilitate to separate the molecules based on electricity, and that will be our electrophoresis. So electrophoresis can able to separate molecules depending upon their electric charge also. So that is the idea of electrophoresis. So this is the electrophoresis. So electrophoresis is a lab technique that is used to analyze molecules. Especially this is extensively used for separating the genetic molecules. Okay, the DNA or RNA fragments can be efficiently separated with the help of this electrophoresis. So there are a few modifications has been made in comparison with chromatography, such as in chromatography we used paper, whereas in electrophoresis we are going to use gel. Okay, in this gel we are going to run the samples, and this gel is called as polyacrylamide gel. Okay, PAG. Hence, since we used page, it is called page, polyacrylamide gel electrophoresis. So yeah, that is the idea behind this electrophoresis. So very simple. You suspend the molecule, the specimen that need to be separated in this gel, and you will apply electricity at both the ends of this gel tank. Okay, anode and cathode. So these molecules, there are certain things that we need to consider. One is the overall charge of this molecule should be negative. So that can be achieved by adding some chemicals to this. I forgot the chemical. I think some buffers will be used to maintain the even charge among the molecules, the DNA molecules. Then we will we will run the sample via passing electricity and they will get separated. So the separation depends upon the size, shape, electric charge, and how it is interacting with the stationary phase and the mobile phase of the separating medium. So by considering all these parameters and factors, we can able to separate the this genetic or DNA or RNA. Okay, so that's it. That is all about this gel electrophoresis. And there is another modified gel electrophoresis called Sutherland blotting. So blotting means now we can able to transfer the molecules that are separated from one gel to another gel. That is called blotting, and that is advanced process. Okay. The blotting we will see later. Anyway, this is all about the electrophoresis. So there are many types of electrophoresis. We have agarose gel electrophoresis. As the name suggests, we will use agarose gel in it. And we have polyacrylamide gel electrophoresis. Here we are using polyacrylamide gel. So there's a difference between agarose gel and polyacrylamide gel. Agarose gel has been extracted from a seaweed. It's a plant. Whereas polyacrylamide is a synthetic gel, which have the similar properties like a agar. Okay, so this is the difference between polyacrylamide and agarose. Then what are the applications of electrophoresis? So one of the straightforward application is separation of DNA fragments. So we will add some probes of our interested candidate, the genetic candidate, and that will be targeted and that will be separated via this electrophoresis. Simple. Okay. If we suspect the patient has some particular problem, maybe we are targeting one particular gene, and the corresponding gene fragments or primers will be used in the PCR. Upon getting amplification, we will add the sample in the gel and we will go for separation. In the starting days, during during non-availability of electrophoresis, we used a traditional approach called Sangus sequencing. So Sangus sequencing is really painstaking. Just for knowing 200 base pair sequence, it needs almost 1 week of intensive lab work. But today we can able to sequence at high speed. Even the entire human genome can be sequenced in a matter of days, actually. So, from 200 molecules to 2 billion molecules, from 1 week to 2 days. So that time, the things has been really advanced, and now it is very easy. And all these things are possible because of these techniques like electrophoresis and PCR. So that is the idea behind this electrophoresis. And not only genetic material, but also we can separate protein candidates, protein molecules in this electrophoresis. Okay, proteins that are very minute, proteins that are not even capable to separate in chromatography techniques can be separated with the help of this electrophoresis. The add-on advantage of electrophoresis is the presence of electricity. So this electricity will give us an additional it will make the molecules to migrate fast. When I say migration, that results in separation. So that is the idea of this electrophoresis. So there are multiple or plethora of applications of electrophoresis. Some include for clinical diagnosis, whatever molecules is suspect that is pathogenic that can be detected in the labs. We can do research, food molecules, environmental studies, forensic studies, cancer studies, even hormones can be separated. That depends upon the modification of the gel and the type of what is that casting we are using. Okay, So that's it. That is all about the electrophoresis. Now, depending upon the shape of the gel that we are using, these electrophoresis are further divided. Is the separating molecule only separating in unidirection, or double, two dimension, or three dimension? So if you are using a paper like a gel, then that will be two-dimensional electrophoresis. If you are using any column, a test tube like column, then that will become a three-dimensional electrophoresis. Okay. And those three-dimensional electrophoresis traditionally called as Western blotting and Southern blotting. So the difference between Western and Southern is one is used for protein separation and other one is used for the genetic molecule or DNA RNA separation. That is all about the electrophoresis. Then let us see a few multiple choice questions such as what is the principle of electrophoresis? So the electrophoresis primary principle is apart from separation based on size, shape, etc. or molecular weight, it can also separate depending upon the electric charge of the separating molecule. That is the major advantage of electrophoresis. So the answer will be B, separation based on electric charge. Then second question is which of the following is used for separating large molecules such as DNA or RNA? Obviously, you cannot separate DNA in agarose or capillary. Okay, you can only separate in agarose gel electrophoresis. Okay, polyacrylamide is used for protein separation. Capillary electrophoresis is entirely different story. That is, capillary electrophoresis is paper electrophoresis actually. Okay, chromatography. And isoelectric focusing is a okay. So isoelectric focusing means it's an approach where the pKi of the molecule. So each and every molecule has its own designated charge, and once the applied external electricity is sync with the molecular electricity, then only that corresponding molecule can be isolated from the heterogeneous population of the molecules. And such technique, such approach is called as isoelectric focusing, and that is really advanced method. And isoelectric focusing cannot be achieved with the traditional electrophoretic method. We need an advanced equipment to do isoelectric focusing. So this is all about the electrophoresis, and maybe we will see next at 2 o'clock. Thank you very much, students. I hope you already wrote your names in the chat box, right? So thank you very much. You can leave. Everyone can leave now.